Perfect. Okay. I should start with a health disclosure statement since I'm wearing a mask today. I've been coughing for like a month, and the doctor says I'm okay. He said I have some kind of inflammation problem, but I should wear a mask just to be safe. But I feel okay other than the coughing on occasion. So that's why the mask. But I'm COVID negative, pneumonia negative, and all that other fun stuff. So I want to thank the IGB team for the invitation. So I actually recognize several of you in the room, and some I don't. So this will be fun to have a fun conversation. I haven't done this before, so I kind of wasn't sure what to talk about exactly. So I have some broccoli stuff, but I also have some food science and food micro stuff to talk about too, just to talk about it all. So, but we'll get to broccoli, don't worry. We have 2/3 of this will be on broccoli. So as was mentioned, my name is Michael Miller. I'm a Angelino, meaning I'm from Los Angeles originally, and from Pasadena, actually real close to the Rose Bowl. Never knew I'd end up in a Big Ten school later at the time, but that's where I grew up. Parents still live in the same house. I won't tell you how much they paid for it and how much it's worth now. It's really obscene. But I came to UIUC in 2006. I'm here with my wife is here, Catherine, and I have twin boys. They're both seniors at Champaign Central. We have a big soccer game tomorrow with the #1 ranked team in the area, the Champaign Central Maroons. Before, I'll just give you a little history about my background, and I don't even think my wife knows all this, but I grew up in California and went to UC Davis for undergrad. Davis is in farm country like here, not like Los Angeles, very different. But there, I had an opportunity to work with this fellow here, Mike Dennison. He was always Mike, never Dr. Dennison. Very interesting guy, kind of funny. I found a picture of him holding a broccoli, so I had to use that today. But he gave me, at the time I had no idea what research was or what a future in research would look like. But he gave me that opportunity to work in his lab. I loved it, and it kind of propelled me to more opportunities. So this always reminds me that it's very important to give those opportunities to the new scientists in the community. And then I went to MSU where I did my PhD with John Linz. So I should mention that Mike actually passed away a few years ago, sadly. Dr. Linz is still with us. He's retired, but he was at Michigan State Food Science Department, Food Science Human Nutrition. I studied aflatoxin biosynthesis there. He was actually a college quarterback in his high school or college days. Very good athlete, really a fun guy and a great scientist. I did lots of aflatoxin work at that time. And then more close to what I do now, I was at NC State and I actually found a picture with me in it. So that's me, I'm the tall one in the back. That's with Dr. Kleinheimer's in the front with the kind of the gray turtleneck. At the time, he was the only National Academy of Science member that was a food scientist. So he was a real big deal. It was great to be a postdoc in his group and learn a lot about lactic acid bacteria, which is the big focus of my research, is these organisms. And I want to, perhaps most of you don't know what food science, human nutrition, like we have such a department here at U of I, and what we do. We have over 30 faculty in our department. We have over 300 undergrads with 4 undergraduate majors, including food science. We also have several graduate programs, thesis, non-thesis programs that are at the moment we have close to 100 thesis students, graduate students in our department, so it's a very active group. And I'll show you a picture of a bunch of them here in a moment. I didn't know what food science was until I was a PhD student, so this is, I just kind of stumbled into a PhD program that was in the food science. But not everybody is familiar, so I wanted to pull some stuff from the IFT, our trade association, and describes what food science is, but it draws from many disciplines: biology, chemistry, engineering, and biochemistry to understand food processes and improve food products for the general public. We have our undergrads graduating to get jobs with like PepsiCo and Kraft and Heinz and all the kind of companies you're probably familiar with. They, the most recent data we have is they're getting the ones that go to Chicagoland or other big cities, they're getting job offers in the 80,000 plus zone for an undergraduate degree, which is pretty amazing. That's more than our starting faculty salaries in my department, which makes it, interesting conundrum. But it's a very lucrative, and the food industry is never going to go away, so it's a very stable, impactful place to be and have an impact. But I'm a food microbiologist, really, so I want to spend a moment about food micro and what that is. I teach a food microbiology class, and we talk about the three pillars: safety, spoilage, and fermentation. Maybe doesn't always appreciate the magnitude of these things, but safety, food safety is probably 50 million cases of foodborne illness in the US every year. So 1 in 6 Americans have foodborne illness every year. That's a CDC estimate, so that's, it's not a rare event. And these have costs. These costs are economy, they cost lives. There's close to over 3,000 estimated deaths due to foodborne illness in the US. So it's a significant problem. Spoilage, microbial spoilage in particular. If you think about food security and the lack of food for the population, thinking about how we can preserve food better, make more food available for people that want it, is very important. Perhaps there's estimates that maybe 50% of fruits and vegetables are not even consumed, are lost due to microbial spoilage. So just think how much better our food supply would be, or our food security would be in the US and in the world at large, right, if we could have a better control of food spoilage. And then more closely to my interest is fermentation. And the fermentation is very important. The fermentation industry is very large. It's not just beer. Many foods are fermented in the US. Sauerkraut, of course, kimchi, things like that, but things, soy sauce, chocolate, coffee, all the cheeses and lots of dairy products. There are many things that are fermented and maybe you don't appreciate are fermented. Hot sauces, all sorts of things. So fermentation is a very vibrant part of our economy and very important. And just if you further convince you, this is a picture we took just a few days ago. This is the entire food microbiology team at a little party we had on a Friday, last Friday, I guess. These are all, except for a few outliers, these are all food microbiology grad students or staff or faculty in the food science and nutrition department. So you can see quite a healthy group, a lot of people. We have 4 faculty. Dr. Banerjee is a food safety kind of extension specialist, does a lot of work with the food producers in addition to doing research. Young-Soo Jin is a yeast metabolic engineer, does lots of yeast metabolic engineering to make new things. I know her. Hi, Lavender. Yeah. Anyway, now I have real pressure on to say Matt's name right. Matt Stashevitz, that's not close. I was doing better when I wasn't being watched, but Matt is also a food safety. So we have 4 of us, 4 faculty in food microbiology in Illinois, which is pretty robust. Most universities don't have more than 2 or 3. We have a very strong group. Okay. And then I wanted to mention in 175, the Science of Fermented Foods, the class that we have that I developed. It's completely online. It's all recorded lectures, recorded videos, tours of like Prairie Fruit Farms, Riggs Brewery, Central Illinois Bakehouse, which is downtown Champaign, lots of places like that. It's a really fun course. We have people from the community take it every semester. So if anybody's interested who wants to do that, you can certainly do that, and you can reach out to me and I can help you figure out how to enroll to do that. We even had people from the public health department in Champaign, Urbana take the class. You can imagine there's lots of people fermenting things and trying to commercialize things, and they're like, well, you don't know what this is, so they took my class to help learn some basics about fermentation. Okay. So we can get into the science stuff. You guys can ask questions too as we go. This doesn't have to be formal exactly. I'm a very informal person generally. So I'm here as part of wearing my IGB hat. So I'm a co-theme leader in MME. This is the microbiome and metabolic engineering. And so I'm very interested in the microbiome and their role in health. So we'll be talking about that today. But you can see on the slide all of the other things I'm involved in. It's not just brassica and broccoli that we'll hear today. I also do projects related to other fermentation fermented foods, also some things related to lactic acid bacteria, the bacteria involved in fermentation, and understanding their behavior better in terms of in foods, but in the gut as well. Okay. And then I wanted to, so a trick somebody else showed me, I like starting with an acknowledgement before I get into the research, so I make sure I thank the people that were involved in the research you're going to see today. And so the picture on the right is my current group. We have Yanling, Zafan, Molly, Adriana, Joshen, Carson, and Devin, and myself on the back. Fawn wasn't there, but she's here today. She was out sick that day. So that's my group on the right. Fawn and Joshen are the ones who are responsible for the broccoli project, and then on the left there's a picture. And she, who's really is in that picture, but you can't see her too good. She graduated, but she's also very involved in this project. And this is all funded by USDA NIFA grants, so we've had a few of those, and this is what's kept this project going. Okay. Another sip. Okay. So brassica. So I'm going to talk about brassica this whole time. Brassica is this group of plants that includes the mustard and cabbage family. Sometimes you'll hear people call them cruciferous or crucifers. That term's kind of fallen out of favor amongst the plant people, and so they like brassica better. Brassica is a more taxonomically appropriate term. And so I'm going to try to use brassica as much as possible today, but examples of brassica are broccoli, cabbage, kale, Brussels sprouts. Think of all these foods that have a little punch to them, these vegetables, especially when consumed raw. And we're going to talk about why that is here in a moment, but that's why these are interesting. When an epidemiologist look at like vegetable consumption and fruit and vegetable consumption and cancer prevention, there's lots of evidence for that space. We should all be eating more fruits and vegetables, of course. But the group that has the most and the strongest evidence is the brassica. There's lots of strong epidemiological evidence that brassica are very health promoting in terms of cancer prevention and other ways. And in fact, there's a lot of mechanistic insight as well through various animal studies. We've learned a lot about how that's possible and come up with reasons and explanations for why that's true. And I'm going to talk about that a little bit. And the key is what makes these brassica unique that only brassica have are these compounds called glucosinolates, or GSLs. And I'm going to call them GSLs and sometimes glucosinolates, and I'll try to keep the acronyms to a minimum today, but glucosinolates. These compounds are only found it's just evolved to exist only in brassica. So they don't they're not in melon, they're not in beef or anything. It's only in brassica vegetables. As you can see in their structure, there's an R group there. right there. So depending on what that R group is, that indicates which particular glucosinolate it is. If you look at broccoli, for example, most of the glucosinolates in broccoli are specifically glucoraphanin. But in kale, you might have gluconapin or bourgoitin. In horseradish, you have sinigrin. These are all different just based on the R group. But the rest of the compound has the same core structure. So it's always this glucose with a sulfur linkage, which is very unique biologically, like that's only in glucosinolates. Okay, so these compounds are interesting, and let's talk about them more. So you got these compounds, they're in broccoli, cabbage, and kale. When you these are stored in the plant, just in the cells. It's also in the plant. They have an enzyme called myrosinase. This myrosinase is kept separate from the glucosinolate. So these two are both present in the cell, but physically separated so they can't interact. If the cells are disrupted, meaning chewed on by an insect or an herbivore, or by you chewing it, okay, you disrupt that cell structure, the enzyme comes in contact with the substrate, the glucosinolate, and you make these ITCs, the isothiocyanates. ITCs, isothiocyanates, are very bitter. This is why when you chew broccoli, it gets bitter. When the broccoli is sitting there, raw broccoli, is not bitter. It gets that punch when you're chewing it, okay? And that's why when you cook broccoli, and we'll talk about this more, but when you cook it, you activate the enzyme, that can't happen. So I imagine many of you probably prefer steamed broccoli, and that would be why. Right? At least one aspect. So that, why me, the microbiologist, got involved in this is because we realized that most people eat broccoli cooked, steamed, and so they inactivate the enzymes. So how are they making these isothiocyanates? These ITCs, I don't know if I mentioned, but those are what we think are responsible for all the bioactivity. The great benefit of brassica vegetables is coming from these ITCs. So we're very interested in these ITCs. But if you eat cooked broccoli, you're not getting any. At least that was the thinking, right? But we realized actually that the gut microbiome, so the community in your colon, can do this too. So that's going to be a theme for my talk today. Doesn't matter if you get it made in your upper GI tract or if you get it by the broccoli enzyme, or if your gut bacteria do it in your colon. Which is better? I don't know yet, but that's one of the big questions, right? That we're thinking about.
So what's happening is a mechanical effect on this molecule that breaks it into glucose and this SCNR thing. Yes. So.
Yeah.
Okay. But do they react with water or something else then? Are they soluble? Is it something, the properties change of these chemicals then?
Yes. So the ITCs are very strong electrophiles. So they get into the real chemistry, but they're very reactive. And that's part of the reason why they're very bioactive, but they're very reactive, and they react with
sulfur in it would be pretty reactive.
Yes. It will stick to free thiol groups, like cysteines, very actively. And it creates, in our cells, it creates interesting pathway that ends up activating this response mechanism. It's hard to talk, simplifying it without saying the words I'm trying to avoid, but it activates NRF2, which is very important. And NRF2 is responding to these kinds of stresses in the cell. Hopefully I didn't butcher that, but these isothiocyanates are like, sulforaphane is probably the most famous one, and that's the one that's from broccoli. And you can buy sulforaphane like probably down the street, the Strawberry Fields probably. Anywhere they sell supplements, they'll have sulforaphane supplements. I don't take it, so I'm not pushing that or anything, but it's because of this, their bioactivity and this reaction, right, is why they're so interesting. The glucosinolates, as they sit, are not bioactive at all. It's only when they're converted to this ITC that they become bioactive.
Mechanical function. It takes mechanical?
Well, it requires disruption of the cells so the enzyme and a substrate come in contact. But it's not mechanical. It's mechanical because you got to disrupt the cells when you're chewing the food. But if I had purified compound and I just mixed with the enzyme, it would work great. But it's mechanical just because the cells they've mechanically separated them.
Breaks the cell and then that gets exposed or something?
Yeah. They keep the enzyme in these granules so they can't do that. They don't want it to happen. They only want it to happen when they're being chewed on by a herbivore. Right? It's a plant defense mechanism that they've evolved. Side against it?
Yeah.
Okay. Okay, so one of our questions, I learned that from my collaborator, Dr. Jeffrey, who was down the hall from me, and she was, Madam Broccoli, is how we knew her, and she was studying broccoli all the time, but I'm just this microbiologist learning on their student committees and trying to learn what's going on, and saw this place for microbiology to be connected to it, and this is kind of where it started. If we fed people, this is a collection of different studies, but like raw broccoli versus cooked broccoli, and look for how much isothiocyanates we can recover from their urine. Meaning they've ate the glucosinolates, it's been converted to isothiocyanates, been absorbed, and been excreted. Okay, so this is the path. With raw broccoli, meaning the enzyme is active, right? You haven't inactivated the enzyme. They chewed that broccoli. They get 25 to 40% of the glucosinolate they ate, they can recover as isothiocyanates in their urine, right? So pretty good yield of that. If we cook broccoli, it's less. You can see it's about half. So you don't get nearly as much that way. About half as much.
Okay, so now.
Yeah.
So what started your interest is there were studies that showed these ITS, whatever. There's been studies that show they have health benefits.
Yes.
Correct? Okay, so then you're studying sort of the mechanism why that is and if it differs between cooked and raw, so you can better understand how those health benefits are achieved.
Yeah. I'll rephrase for you.
Okay, yes.
Yes, because what's interesting to me is we have this compound that's very interesting that the gut bacteria can make, but it doesn't make very well. Actually, I'll show data in a few minutes. It varies a lot from person to person. So now there's a big opportunity. Can we make it so people make more? Doesn't matter if it makes it more in the colon versus it makes it a lot in the mouth. I don't know the answer to those kind of questions, but that's where we're going. That's the plan. This is the first experiment I was involved with related to broccoli. I should probably have like a trigger warning, but we're going to have some animal experiments data to show. This is all with rats and mice. But in this particular case, we injected glucoraphanin, the glucosinolate from broccoli, into the cecum of a rat. At the same time, we're collecting from the mesenteric vein, so this is all the nutrients being absorbed out of the cecum of the rat. We're collecting samples from there. And when we do that, we can see that when we inject glucosinolates in there, if we wait long enough, we will see these isocyanates being produced by the gut bacteria and being absorbed. This was very exciting because we're like, okay, we're proving that the gut bacteria can do it. We don't know who's doing it, but we know somebody in there is doing it. So this was probably in 2010, maybe I did that.
What's a cecum?
It's where the fermentation happens for a rodent. It's like their colon. So it's like a bag between the small and large intestine. We kind of have a cecum, but it's not like that. For them, the cecum is a very important fermentation vessel. Thank you. Good question. Okay, so now we're hitting the data stuff. Okay, so our first question is, okay, so we're going to take these rats, we're going to feed them broccoli, 10% broccoli in this case, and we're going to take samples every so often from them, and we're going to see, does the microbial community change, and does it change in its ability to make isothiocyanase? So on days, so we fed these mice in this case rats, broccoli for 14 days. As you can see at day 0, you see a very small bar. Right? And that's because at day 0, they've never seen glucosinolates before. They don't have any bacteria that are good at this conversion, so they don't make any. Okay? But as they eat more and more broccoli, the community is changing, is increasing in bacteria that are able to do this. And by day 4, you can see they're making a lot of the maximum. Right? So they're kind of goes up and then it hits a maximum. So they're like, okay, so feeding broccoli for 4 days, that's going to be important for our studies. So we did another 4-day experiment. But this time we compared the no broccoli group to 10% cooked broccoli in red. The green one, that's when we extracted glucosinolates from the broccoli and feed just that. So it's matched for the same amount of glucosinolates, but no other broccoli components, hopefully. And then the purple, we've taken the broccoli and we've hydrolyzed all the glucosinolates ourselves and washed them all out. So there should be no glucosinolates and no ITCs in that treatment. But there's still fiber, like, you know, it's still green, right? It still looks kind of like a broccoli diet. So, to summarize this figure, only the groups that fed glucosinolates see the change. So the cooked broccoli group and the GRP group, you can see after 4 days they get more. It makes sense. The hydrolyzed group, at the end, not so much, right? But they're not being exposed to glucosinolates, so they're not selecting for glucosinolate consumers or ITC producers in that group. I'm not showing you all the microbiome data. We have lots of microbiome data, but the big conclusion from that is that if you looked at the control and the GRP groups, they're indistinguishable. But here, they're very different. So functionally, they're quite different. But taxonomically, when we sequence them, we can't see any differences. And it's because the changes that we're seeing that result in this activity change are probably very small members of the community. They're very important in this act function, but they're not, they're getting washed out by all the other changes happening from fiber and other things that are happening in this, the differences in these treatments. So the control and GRP look very similar, and the cooked broccoli and hydrolyzed cooked broccoli are indistinguishable from a community perspective. They're like, those two cluster really good, these two cluster really good, but they don't cluster together at all. It's kind of interesting. Our dream would be, this would help us identify the magic bacteria that would be useful because they're only enriched in the GSL groups and not in the other groups, but we did not find such a thing. It's never easy. And then we have this, yeah.
Have you sampled for each day or is it after 4th day?
We've mostly only sampled on day 4. We've done more sampling on other time points a few times, but it just ends up being, day 4 ends up being the very important day for us.
Yeah, the transient population might be getting up in one of those days. My suspicion would be that could be one of the things that, and then they're kind of winning down and it's been produced. You know, you're looking at the, you know, GRP.
Yeah.
At the 4th day, that's the product.
it's a good question. So it might have been more obvious, like the microbial differences might have been more obvious on day 2 than on day 4, that we've kind of missed it on day 4. Yes, that's possible. I'm looking at who should be thinking about it over here, my students. These are all cooked broccoli, but we did raw broccoli as well once. This one's fun because we did 4 days on raw broccoli, and then we took broccoli out of the diet to see what happens if they stopped eating raw broccoli. So the first thing I want to point out is when you eat the raw broccoli group, we see the same thing. You're probably wondering, how is that possible? Well, it turns out even in raw broccoli, the enzyme works and makes a lot of ITCs in your mouth, but a lot of the glucosinolates still escapes and makes it to in the cecum of the animal. So you still get so when you eat raw broccoli, you're getting both events. You're getting stuff made upper with the enzyme, but then you're also getting stuff by microbial community in your gut. Both are happening. And in this case, when they went back to control for 3 days, it approached the lower level of the control. So it kind of goes away. So ebbs and flows with how frequently you're eating it, right? At least in terms of the rodents. So we have some human data to support this as well, but not as robust as the animal data. Okay, so frequency of consumption is important. Eating it more often is going to result in altering your community to be better at making isothiocyanates. That microbial change is going to be true for cooked or raw. And that you need to be eating, it's the glucosinolates specifically that's causing this change, not other parts of the broccoli.
So when you talk about cooked, are you talking about microwaved or boiled or fried in a wok or what?
All those. So anything but very lightly steamed would inactivate the enzyme. So the enzyme's pretty easily inactivated.
Breaking the cell, you're saying.
Well, it's the so if the enzyme is heat inactivated or microwave inactivated or something like that, it doesn't matter what happens after that. All the chewing or breaking up the enzyme's dead, so it won't do it. So then it has to rely only on the gut bacteria to do it. My colleague, Dr. Jeffrey, has done a lot of work figuring out what's the best way to cook broccoli and still manage to keep the enzyme. It's possible, but you have to steam your broccoli so that it's still crunchy, like really crunchy. So I think most of you probably steam your broccoli so it's like at least lost a little bit of that firmness, then by that time you've inactivated the enzyme.
That's mechanically breaking down the cells.
It's not the cell. It's not breaking down cells. It's like the enzymes will denature with heat, and once they denature, then they're not going to be functional. So it's still there as a amino acid, but they're not going to have that enzymatic activity anymore. Good question. One of the funs of being an advisor all these years is you get slides from your students, and your former students in this case, and you can see their personality in their slides when you steal them and take their defense slides and use them yourself. And so I could see Anchi saying these things as I pull these slides out. It's kind of fun, but let's take a step back. This is in Anchi's words. So I've been I've kind of, I don't want to say lied to you, but I've been very narrow in thinking. And that I've been talking only about the glucosinolate to ITC conversion, which is this step here. But in fact, we've learned that there's lots of other metabolism that can happen with glucosinolates. And all these other things, like making a nitrile, for example, these are not bioactive at all. And so all these other things are pretty much unwanted if you want to make as much ITC as possible. And so we were nobody had really looked at these other metabolites in the gut, what gut bacteria are making it, how is this important. Now, if you think about it, you have this product, this chemical, and it can be metabolized many different ways. Only one of those ways is to making something very bioactive and health, cancer preventing, and this kind of stuff. The rest of them are not. So you want to minimize the flux to these unwanted products and maximize the flux to the desired product, right? That's how I approach this when I think about this problem. And I think about each of us as a walking fermenter, right, with the microbial community in the gut. Can we maximize making more ITCs, right? This is kind of where I'm going with this process. So the isocyanates, of course, but these other compounds like the desulfated glucosinolates, so these nitriles, are also very interesting to us because these are metabolites that are taking away from the ability for us to make ITCs. And so we were very interested in that process, and so we were wanted to test that question. So how does broccoli consumption impact the microbial glucosinolate metabolism, not just ITCs, but global metabolism of glucosinolates? We made this fun figure for the paper that was submitted. This is actually one of those times where you get to use the figure one of those figures called you make for the like table of contents figure. Graphical abstract, yeah. This is actually using a graphical abstract for something useful. Most of the time you do those and it just goes in the paper, but you never look at it again, and you're kind of annoyed that you have to do it in the first place. But I grabbed it and used it for this. But in this case, we have lean mice, fat mice. We're feeding them a low-fat diet with and without broccoli, or a high-fat diet with or without broccoli. And we're looking at how this is changing their microbial community, how is this changing the community's metabolism of glucosinolates. That's what we're going to get after here. That's not interesting. So let's talk about this. So in all cases, you can see in the green boxes, when they're eating, so if just look, like let's do the top left box with the lean animals. When they're fed the low-fat diet, we don't see any ITC production from those groups. But from the ones fed low-fat cooked broccoli, we can see lots of ITC production when we feed extra broccoli to that group. It's true for some of these sulforaphane metabolites that we looked at, and even the nitrile. We see a trend that is higher in the cooked broccoli group, but wasn't statistically significant with the number of treatments that we had. But what's cool is what you could do from an experiment like this is you can start doing these kinds of analyses where you can look for how different particular groups of bacteria correlate to different results. And you start looking at things like, for example, Blautia, which is in the third one there. You see as Blautia numbers increase, the amount of ITCs goes down. So Blautia is probably not making ITCs, is preventing the ITC makers from working or taking glucosinolates and making other things and reducing the ability of the community to make ITCs. So we can use this kind of correlation and association data to start testing hypotheses and developing new experiments to kind of test out which bacteria are responsible for which activity. The major challenge in this space, I should mention since Josh Chen is here, is measuring all these stupid compounds. So measuring these nitriles and the desulfated glucosinolates and these glucosinolate metabolites, isocyanate metabolites, is no small task, and it's been a fun process for Josh Chen to be LC expert, but she's making a lot of great progress. Okay. So we want to identify these microbes. Can we use that to explain why some people make a lot of ITCs and other people make very little? My hypothesis, the people that make very little, is because they have bacteria that are very good at making nitriles. And these other, their flux is going to these non-sulforaphane producing, ITC producing pathways.
Have you tested people's biomes?
Yes. We've done some of this with people. In terms of with people, what I could summarize with people, we've been involved in a few studies where they fed either broccoli or kale. It was either 250 grams or 500 grams a day for 2 or 3 weeks, which is a lot of kale, actually, 500 grams a day. It's pretty obvious what group they're in when you get their fecal sample, actually. Yeah, so not as blinded as you would think. But they, yes, some people are very good, their community is very good at making ITCs. Some people are very bad. After 2 or 3 weeks of kale consumption or broccoli consumption, some people become very strong. Like you can see a very good response like we saw with our rats. And other people stayed low, and the people who were high stayed high. So we have responders and non-responders in this kind of experiment. So I think this is going to be very relevant for people as we go delve more into that space.
Why do you use obese lean mice?
So we used obese and lean mice because they're yeah, so this is a long-winded answer. It's because the next study I'm going to talk about has to do with GLP-1 and glucose control and diabetes and stuff. And so we want an obesity. And for our grant proposal, we had to show that what we showed for lean mice is also true for obese mice because we're using obese mice in the next study. So it wasn't because we had a great idea at the time, it was actually forced on us on a review panel. You'll see why in a moment. Yeah, review panels always have good suggestions. Most of the time. So, all right, so we have, as I kind of hinted at, these ITCs, very bioactive, very exciting for cancer prevention, lots of other things as you can see on the list. Most of this is mediated by this NRF2 pathway, which is a very important pathway in the host and all of us in terms of reacting to some of these stresses and mitigating these kinds of issues. So NRF2 is a very important pathway. But I wanted, we had this idea that maybe NRF2 isn't the only reason why these ITCs are important. And I want to talk about why bitterness is important. So if we think about ITCs, so when we've all experienced this. If you consume raw broccoli, raw kale, all the raw brassica, very bitter, right? At least, maybe you might not use the bitter, but very pungent, very strong tasting. That's because as you chew the plant, in the raw plant, you're getting a lot of ITCs being produced. These ITCs are very bitter. This is why the plant makes them in the first place, because the insects hate it. They go, this tastes like garbage. No, thank you. I'm gonna go eat something else. Right? This is the whole premise. And so when we eat the raw broccoli sitting there doesn't taste like this. It's as you're chewing it, that chewing it is mechanically separating the enzyme from the substrates, allowing this to happen, and you're making this bitter ITCs, sulforaphane in broccoli's case, in your mouth, and you're getting hit with this bitter compound, interacting with the bitter taste receptors in your gut. When you eat cooked broccoli, this cannot happen because the enzyme is inactivated. So that's why cooked broccoli, like at least anything other than lightly steamed broccoli will have this issue. You inactivate the enzyme, that's why it's not nearly as bitter, right? Cooked broccoli is much milder. But then, when you think about the whole digestive tract, in the colon, you have the if you eat raw broccoli, you're going to make bitter stuff in your mouth, but then some of the glucosinolates is going to escape, make it all the way down to your colon, and you'll make some ITCs in your colon as well. If you cook broccoli, you're not going to make any bitter compounds in your mouth, but you're going to make a lot more potentially in your colon, right? So it's a very different situation. So we're like, is that important? Like where it's made, how much is made, how is that important, right? So we're very interested in this question. And second aspect of this, it turns out you have bitter taste receptors all over your body. They're not just in your mouth. They're actually all the way down your GI tract, they're in your lungs, there are lots of other tissues that have bitter taste receptors. In fact, you have over 25 different ones. You have many, many bitter taste receptors. Now, it's a little bit of a, bias. We shouldn't call them all bitter taste receptors because only in your mouth are they bitter taste receptors. The rest of your body, you're not tasting anything. They're responding to stuff, right? You don't tasting bitter in your colon. But it's the same protein, same receptors responding to similar cues. Okay, so we have, and similar, you have these sweet taste receptors all the way down your gut as well. But I would rather talk about the bitter taste receptors. So these bitter taste receptors are interesting, and I think this is fun for those of us into biochemistry, but what's important, I think, for you is the involvement of GLP-1. So when these bitter taste receptors are activated, such as the T2R in this picture, it results in GLP-1 release. GLP-1. I don't know if you guys all know GLP-1. GLP-1, like Wegovy and Ozempic, a lot of these, you might see their advertisements on TV all the time. These are all, GLP-1 is like, I got involved in this study with maybe, 3-4 years ago before I realized what was coming on the GLP-1 front. But all these GLP-1 drugs are used for weight loss now. So all the Ozempics and Wegovy's, these are like taking a GLP-1 that stays active for a long time. So then you inject it, and then you don't eat for a week because you've had this elevated GLP-1 all the time. Right? This is how those work. And it's your body's way of responding to eating, right? As soon when your body senses these nutrients, it triggers GLP-1, tells you, okay, you've ate, slow down, stop eating, this kind of thing, right? Works with satiety, glucose control, things like that. And so we're like, well, that's interesting, right? If we're can we get ITCs made in the gut by the bacteria, triggering the bitter taste receptors in the colon, causing GLP-1 and making us eat less or have better glucose control, right? That's kind of what got the grant funded, I think, was that kind of idea. And so I'm not going to show any animal data because we don't have that much yet on that front, but I'm going to show you some cell work. So in this case, we took mouse enteroendocrine cells. These are like the kind of the hormonal cells of the gut, and incubated them with these different compounds to see if they make GLP-1. So if you look at on panel A, when we incubate the cells with AITC, this is the ITC from horseradish, from Sinigrin. You can see we can get, the white is the empty bar is the no treatment, but the purple ones, those are different amounts of this AITC. We can get an increase in GLP-1 production. Okay, cool. On the right panel is Sinigrin. That's the parent compound for AITC. So that's the glucosinolate. It does nothing. It looks the same as control. So you can see there's a difference between the glucosinolate does nothing, but the ITC, its related ITC stimulates GLP-1. We then tested several other ITCs. PITC, which I think is that one's from watercress, if I remember right. don't remember where BITC is from, but sulforaphane, SF is the one from broccoli. They all stimulate GLP-1 in our cells at very low concentrations. You can look at the mechanism, and this is like how we function as scientists. Like, this is great, yes, but how do I prove that this is through bitter taste receptor and not just some other response? Like, this is kind of the challenge we have with these kinds of things. And so we look at the pathway, we find inhibitors for the different parts of the pathway, and you can kind of start to prove that bitter taste receptors specifically are responsible for this that we saw.
So do you simulate these chemical pathways in the laboratory?
Kind of. These are with cells, these are mouse cells in culture. And so we can then treat those cells with, say, U73122 or 2-APB, the compounds in the red boxes. Those inhibit those particular parts of the pathway. And we can see how those different inhibitors impact our, in this case, calcium flux, which is, we don't need to get into, but we can talk about this part more. If we look at panel A, control, no GLP-1. Cells treated with AITC, lots of GLP-1, strong induction. Okay, we already knew that. If we treat it with all those three inhibitors, in all three cases, we see reduction in GLP-1. So all those pathways are involved. And it's true for sulforaphane as well. So if we inhibit these other pathways, whether it's the phospholipase C or the IP3 or the TRAPA1 channel, it all results in the same thing, reduction of GLP-1. So we think the T2R and TRAPA1 pathways are both important. Okay, but this is like, this is just a cell culture. Like, it's good for us to understand what's happening, but it doesn't prove it's happening in a person, in an animal, right? In a whole complicated system. And so we were very interested in translating this into beyond just cells. You can, I've kind of left out some things to think about, but you have over 25 different T2Rs. Some, that are in your mouth, 20 different alleles, 25 different genes, and then which ones are expressed where along the length of your GI tract is unknown exactly. So which ones are responsible for recognizing the ITC from broccoli versus the one from horseradish versus the one from cabbage, unknown. And so we're trying to piece some of this out. I mean, the dream scenario would be the ITCs, like sulforaphane recognizes a bitter taste receptor that's very abundant in the colon. That would be great for me, research-wise, right? But in fact, it might be, it's only high in the stomach. That means raw broccoli would be much better for that situation, because the ITC made in the colon is not going to interact with, because it's not expressed there. So we're sorting this out, and the cell culture helps us do that. And so we're going to be doing this cooked broccoli, raw broccoli, looking at bitter taste receptors along the GI tract, and looking at GLP-1 secretion and showing that we can do this. In fact, we have some evidence showing that when we feed broccoli to these animals, they eat less than if they don't have the broccoli. And it's because, when they're already obese, it's because this, well, it could be they don't like broccoli. That's the problem with our experiment at the moment. Or it could also be, they don't, this is triggering GLP-1. So we're trying to sort that conundrum out, right?
But even with your cooked broccoli, don't you have this time lag? Whereas raw broccoli, there's an immediate ITC production. With cooked broccoli, you need this 4 to 7 days to make sure that there's microbes to do the process. Aren't we the same? We should, if we're going to eat cooked broccoli, we should eat it 4 or 5 days in a row? So
that's a great question. I try to get funding to do that kind of question and haven't been successful yet. But I think, yes, the question is, So in animal studies, we're always we have to kind of hit things with a big hammer because we're trying to do things in a short time with big effects. But in reality, we are years of consumption with small effects adding up over big time, right? So I can't do an animal experiment for 10 years. That's not going to happen. But so we kind of, so I've kind of gone off on the rails a little bit. But yes, so a shorter, if you ate broccoli 2 days, took a couple days off, and then had it for a day, and then had kale the next day, this kind of thing, I think that would be perfect. But what the specific recommendation is, I can't tell you yet.
But the best is to eat it raw, still, yes.
Unclear still.
I'm back to the title of your talk.
Yes. So, I mean, that's what we're asking. But, I mean, so it's further complicated because if you eat raw, you're going to get more. But the more is going to be a strong pulse in your upper GI tract, like in your mouth and esophagus and stomach. It's going to be a very short-lived, very high peak. And then what left gets to the colon, and it'll make a little bit in the colon. If you eat cooked broccoli, you're going to get none of that in the mouth, and you're going to get a lower, but more sustained production in your colon. So it's a slow build up, production, and that's where these bitter taste receptors are. So could it be low sustained is better than a quick short peak? And where it's made, how's that met? So we're still sorting. We know it's different. What's the difference in terms of the benefit to you, the consumer? That's the part we don't have figured out yet. Not enough for me to declare one or the other a winner. But I think either way, you should eat it more. Eat more, right? Eat more kale. All right, so eat more brassica. And I think what's coming is going to be personalized nutrition type of recommendations based on the genotype. Like, as you might know, some of you are more sensitive to bitter compounds than others. You might know people that can't eat anything bitter because they're very sensitive to bitter things. These are what the impact of that is in terms of the health benefits of broccoli for that person is of interest to us. How their microbial community is so important in this process, and can we make specific recommendations for people? As I mentioned, we know some people, even if they eat broccoli all the time, their community doesn't change to the point where it makes a lot of ITCs. And so it's like, what can we do about that? Can we make some personalized suggestions for them? So I think that's what's coming. I've kind of been vague too, but a lot of our work's with broccoli, but kale and these other ones, other brassica have different glucosinolates that react to different bitter taste receptors. And so it's not clear that they're all going to be the same. So that's next grant, I guess, that kind of stuff, right? Always more questions. Yeah, ask.
Thank you for the talk.